And with that, it's a great honor to introduce Connie Jimenez. Connie is a full professor of translational oncoproteomics from the Amsterdam University Medical Center in Amsterdam, and pioneering already since her PhD on mass spectrometry, all kinds of applications, predominantly in oncology, but more and more moving into a multi-omics space. So it's a perfect kickoff for today. So Connie, please.
Good morning, everyone. It's my great pleasure and honor to be here talking to you at this 7th anniversary of the X-omics Festival. So, cancer is a very complex disease. It's also multifactorial because it results as an interaction of our environment, our diet, our lifestyle, in interplay with our genetic vulnerabilities. And especially for the young people here, if you don't want to get over here, we live in the age of tsunami of chronic diseases of aging. If you don't want to get here, think about your exposures, your diet and lifestyle. If you clean that up now, you may have not have to deal with a disease like cancer. And there's one important factor, which is chronic systemic inflammation, that is linked to all these diseases. And also in the cancer field, cancer is typically regarded as a genetic disease. That's like the traditional view, but this is now it's known it's more complex than that. And there's a big acknowledgement of all the other factors, because the effect of environment, diet and lifestyle is huge, as this creates a permissive environment for cancer cells to be selected and grow. And of course, this is a very complex thing. Once we need to deal with cancer, a multi-omics approach will get us a better view of the biology, and thereby it may enable better treatment options. I also want to make the case that a proteome, with the proteome, you don't get a simple piece of the puzzle. Actually, the proteome integrates all the different aberrations that can occur at multiple levels, at DNA, RNA level, but it also integrates the effects arising from our environment. And this can all boil down to altered activity of signaling pathways. So by having the proteome, this very important layer of information, this can give us an improved understanding of the cancer complexity, its mechanisms, and it will ultimately, and I hope to make this case also in my talk, that it can enable to do a better job at matching treatment to the right patient or the tumor profile of the individual patient. And another point to make, of course, is that most of the targeted therapies of today target proteins. So this really makes the case for including the proteome and also the functional relevant phosphoproteome in the multi-omics toolbox. Before I get into our own research, I would like to highlight also some important multi-omics cancer studies at the international level. And one great effort has been from the cancer, from the CPTEC network. It's a consortium in the United States that received, I don't know how many multi-millions dollars of funding from the National Cancer Institute, and it was formed to accelerate the understanding of the molecular basis of cancer through the application of large-scale proteome and genome analysis, and they coined the word proteogenomics. And as you can see in this picture, The network, I think, has been going for 20 years, but in the last decade, they really made a very distributed network with specialized functions for getting the samples, doing quality control, data generation centers, data analysis centers. And so this is a huge effort, and this effort paid off, especially in the last decade, where there was now about 11 or 12 cancer proteomes have been profiled in great detail with numbers at least 100, 150 tumor samples per study and analyzed in a multi-omics context with DNA and RNA data, and sometimes also phosphorylation data or other types of data, glycosylation. And there is this is a picture from a nice review, like what can we do when we do multi-omics, what can be learned? Well, one of the analyses that was pioneered by this consortium is to use the mass spec data and search it not against the general FASTA files of sequence FASTA files, but against customized databases that include the cancer variants. So this is the proteogenomics definition, let's say pure sang, where that enables to really identify variant peptides and variant proteins that are really tumor specific, either can be used as biomarkers, neoantigens, drug targets. So this is but now it evolved more into the, let's say, multi-omics. You do this different data flows and either do an integrated analysis or analyze them separately and stack them on top of each other. But of course, this is a very interesting if you want to make specific markers or find new antigens. And another analysis that has been done in all these studies is to do a gene dosage analysis where you look for signals that are, for example, amplified genes, lead to overexpressed RNA, lead to overexpressed protein, and maybe also overactive protein. And this can prioritize genomic findings because, of course, there's a sea of mutations. It's hard to distinguish causal features and passenger alterations. So by having multiple information layers, this can prioritize cancer drivers and drug targets. And then at a third type of application can be to get a better understanding of biology, to look at all kinds of features that are either regulated at the post-transcriptional or post-translational level, and also infer functional biomarkers and druggable events. So to highlight the gene or how to prioritize when you have multiple information layers, this is one picture from the first paper from the CPTEC network published in Nature, and all these studies, they're all published in Nature, Cell, Cancer Cell. Here we see the copy number aberration messenger RNA correlation. So this is colorectal cancer, and it's known to have at certain chromosomal areas like 20q that there is massive amplification. Multiple copies, and this results to many overexpressed RNA molecules at many different chromosomal locations. And this is the same analysis, but then for copy number aberration protein analysis. And again, here we see the 20q gain, where that leads to a lot of changes in the proteome. And then by correlating DNA to RNA and to protein, one can see the ones that are really highly correlated, because in this 20q area, there's many genes, so which one are the driver ones? 20q gain is associated with cancer formation. Here we see that the gene HNF4 alpha is correlated across all the levels from DNA to protein, whereas this gene is not. So then the hypothesis was that HNF4 for alpha can be a driver of colorectal cancer. And indeed, this was then validated in vitro by analyzing large-scale analysis from the Achilles project, where it was shown that colorectal cancer cell lines are especially sensitive to knock out this candidate driver gene and not the other cell lines. So this kind of validates the approach and this kind of analysis and why multi-omics can add value. So we took this same approach in another study in colorectal cancer, but then asking the question to look at the adenomas specifically, because adenomas are the precursor stage of colorectal cancer. And by integrating whole genome sequence data with RNA-seq proteomics and then verifying by histology, we could come up, we divided the adenomas in high risk and low risk. And then by overlapping the different analyses and the different differential genes, we could come up with a selection of top 3 genes or proteins. And here you can see the data at DNA data, RNA, protein for the 3 proteins. And you see very nice trend profiles increasing from normal to low risk, high risk cancer. And it's specifically an event of the microsatellite stable cancers or the chromosomal unstable ones. And in this way, we can prioritize candidates for follow-up. And this POVID gene was also validated at the immunohistochemical level, so potentially giving us a biomarker that can indicate adenomas at high risk of progression. And it's a protein involved in notch signaling, so this is an important signaling pathway in colorectal cancer oncogenesis. So in later years, the proteogenomic analysis of this CPTEC consortium became more elaborate and all kinds of analyses were added. For example, the analysis exploring immune landscapes by using different tools, sometimes combining with single-cell cytoph analysis, but also by using deconvolution tools to come up with more elaborate analysis of the bulk tissue proteome profiling data in this multi-omics context. So this is another nice review to look up if you're interested. And in the last year, so then there was the collection of about 11 cancer types profiled in great depth. And in the last year, pan-cancer analyses were published from the same network. And here you see some of the titles. There was a whole special issue also in Cancer Cell. So data, it's all about data integration and then even getting more insights of what features are shared among cancer types and which ones are more specific. And this is one paper out of this series that is, I think, is of specific interest because it is a great resource where they prioritize therapeutic targets, not only by integrating the tissue data, but also integrating it with cell screen data from the DEPMAP project and other large-scale data resources for drug targets and so on. And they have a few great studies in there where they also functionally validated some of these predictions. So with this study, they went from, let's say, purely describing cancer biology in great detail to really getting out actionable findings that were then validated in vitro. I also was asked to share a little bit about the PIHub project in China. PIHub stands for Proteomic Navigator of the Human Body. And it's a huge, large-scale proteomics project in China that was officially kicked off last year. And it has received now, so it is supposed to run for 30 years. And the first phase of 10 years has received 5 billion of yuan funding, which is about 600 million euros. And I'm on the advisory board of this project. I know a little bit more about it, and I got some slides to share. So there was, end of last year, a white paper published in Nature, of which I'm co-author. And it's to summarize, it's a mission of PyHub to digitize proteomic spaces across the human body, individuals and populations, as the basis for building a computational model called the PyHub Navigator, which will enhance our understanding of human biology and facilitate precise disease prediction and prevention. So that's a great promise, and of course this will not happen overnight. It's a big consortium, will have run for 30 years. The idea is that as much money will go in here as once went into the Human Genome Project. There is also, they like there's international labs involved, mainly in advisory role right now. And it involves many groups in China and a few core labs. And in Guangzhou, there's the headquarters. I visited there last year and the year before, and they built a huge campus on an island in the river there. And many machines, and here you can see the performance of these machines. So we are at 8,000 proteins within an hour, typically. Great reproducible data. So I want to make the case, this is also our experience, technology is ready. We are ready. It's a matter of applying now. It's a matter of having large-scale funding to do large-scale studies and make clinical impact. Because that's what we need now. It's large-scale application. We have the tools. And this is an image from the white paper. So there is basically 3 phases envisioned. The first phase is to map out all cells and tissues in the human body, the proteomic anatomy space. The other goal is to map out the states, the physiological states or disease states, and also developmental stage, so it's really big. And then the third part is the proteomics-driven navigation, which is more about computer models and prediction tools to predict disease or how to prevent disease. And so the first goal is all about mapping single cell or cell type resolved proteomic measurements. The second one is about population scale measurements. So we need high throughput technology, but we have it, and we need robotics, and we have them. And then the last part is about cutting edge computational technologies to make a virtual space termed meta Homo sapiens. And I mean, they are gearing up. They are already doing so many studies, not only in the thousands of samples, but even more. And if you are interested, you can read more in the white paper that appeared in Nature. Okay, so now I get to some of our own research. Phosphoproteomics of cancer, and I try to emphasize the multi-omics context of these studies, some of these studies. But first, a little bit more to step you through the toolbox. I'm not sure how many of you are actually doing proteomics. So when you do cancer proteomics and clinical cancer proteomics, of course, the collaboration with the clinical colleagues and the people in pathology is key to get the right samples that you have actual tumor in your sample, that you know what the tumor cell percentage is. And of course, we also do a lot of biofluids, that they are collected with standard operation procedures. Also the tissue for phosphoproteomics, very important, that it goes into the liquid nitrogen or the -80 fairly quick. And not that it doesn't sit overnight, for example. And of course, we need unbiased study design, and then we lyse the tissue, get the proteins, digest proteins to peptides. And if we want to do phosphoproteomics, we do a selective enrichment step for that we automate it. Then we separate peptides by nano-LC. We measure the peptides by mass spectrometry. That's our central tool. And then, of course, we have all kinds of data mining, QC plots to verify that the measurements were correct, heat mapping, unsupervised data overview, supervised analysis, statistics, etc. So it's also a multidisciplinary effort, and you need all these people that can do this in the team. So we use a special flavor of mass spectrometry, which is called data-independent acquisition mass spectrometry. That is let's say, it kind of combines the best of both worlds. It combines the best of shotgun proteomics, or really global proteomics, with the more, that is global, and the more quantitative precision of targeted MS. Because all the peptides are fragmented that are in a certain mass window, so that takes out the stochastic sampling issue. And we were part, some years back, on an international benchmark study where we really showed all labs in a 24/7 modes that data acquisition is very deep and is very reproducible, and also there was some spike-ins with, let's say, ground truth for quantitation, and all labs had a great performance, including our own lab. So with especially data-independent acquisition mass spec, that's the important tool that we see to be applied. So it's all label-free. Also in China, they all focus on DIAMS, all label-free. So here we also, DIAMS works very well if you have deep spectral libraries. It's not a requirement, but it helps to have disease-specific or tissue-specific spectral libraries. And we also, together with Chanan Guo's lab in China, we created two very deep libraries also for the community to use. And also my lab has been involved a lot in getting the right statistic or dedicated statistics, data processing. We published all kinds of tools that we also share online via the repositories, or make it available via website as well. So there is, but I think we are done with the tooling phase. We can apply, and one application that I just want to highlight, summary, is also a recent study that that we did, or actually it has been running for, let's say, the past 6 years, but it's now almost accepted for publication. And we created a large pan-cancer landscape in a collaborative effort with 4 labs, especially the lab of Chanan Guo in Westlake. He's one of the key PIs now of the PI-Hub project, and our lab. We contributed most of the data, and we created the landscape to get a better understanding of cancer biology, to identify targets and biomarkers, and also to develop a pan-cancer classifier to classify primaries or cancers of unknown primary. And all the data is made available in a queryable data portal. And we used, of course, DIAMS with this, because this is the only way we can get high throughput within 2 hours, and currently 45 minutes with the latest instruments, to get deep cancer proteomes in single shot. And this is the summary of the paper. So we defined top cancer type enriched proteins that can either be used as biomarker or drug target. We did a co-expression analysis across the whole matrix to define shared signaling hubs and highly connected genes as potential targets. We also did a multi-omics. It was RNA and protein analysis on colorectal cancer. We have a whole use case defining subtype protein markers, looking at their, and combining it to immune type subsets with prognostic value. And the last application is to create the pan-cancer classifier to classify metastatic tissues. And just to show you that even a large-scale effort across several labs, even several sample prep methods, with robust technology and robust reproducible protocols, it can be done and it can be valuable. So here we see the unsupervised protein matrix clustering, and here we have unsupervised the clustering of the hallmark score with single sample gene set enrichment analysis. And what I would like to point out is that, well, we get largely cancer type clustering, but importantly, this cancer type clustering is independent of the lab that acquired the data and largely independent also of the sample prep, whether it was fresh frozen tissue or formalin fixed paraffin embedded tissue. The prostate cancers still cluster together, or here the liver cancers still cluster together. And here we can see like the signatures enriched, for example, for liver. We get biology that we recognize as liver biology, like xenobiotic metabolism, or for prostate cancer, androgen response. So we are really in business with this large proteome data matrix. And what I also like to point out is because single cell analysis is getting popular, but even when you have bulk tissue proteomics data or bulk transcriptomic data for that matter, one can pretty well deconvolute the different cell subsets in there by using tools. Actually, we used a tool from the RNA world applied to our proteomics bulk tissue data that works very well. And this is kind of the benchmarking to show that the T-cell subsets are enriched in the T-cell leukemias. The B-cell subsets are enriched in the B-cell lymphomas, etc. So this is kind of rediscovering the ground truths, and one can then use it in single sample gene set enrichment mode to explore the different subsets in the different tissues. And then we see that the cancer type of a tumor not fully determines its immune infiltration pattern. There's also within cancers heterogeneity of the immune infiltration patterns. And of course, this can be very relevant for whether immunotherapy will work or not, because with this we can also define, let's say, hot tumors and immune cold tumors. So we were excited to see that this can be done, or we can use the tools that are out there. And we applied many tools, and then some work better than others, but that will be in the manuscript. And the last part was because we have this large pan-cancer landscape, to make a classifier. And this is the top graph, is the performance on the classifier on the left out set. And you can see it works quite well. Only some cancers are not so good classified, but for the majority it works well. But more importantly, we had 2 external data sets of kidney cancer and breast cancer that we could also, they were DIA data sets that we could also classify very well. And we also had, this was the real challenge for us, the classifier has been trained with primary cancer data to see if we can also correctly classify metastatic tissues. And we used this on metastatic tissues of colon cancer and ovary cancer where we know the primary. And we can see that it works very well. And now, of course, it remains to be used in practice to get the primaries or metastatic tissue primary unknown, and then benchmark it against the genomics classifier that, for example, in the Netherlands, Hartwig Medical is using. So now I get to phosphoproteomics. So this has been my lab over a decade has put work into that, and of course a lot of oncogenes are kinases, and we have altered signaling pathways. So it's not, because every genome is so different, so many aberrations, it's not straightforward to know which drug to optimally match based on profile. And of course there's many kinase inhibitors, so phosphoproteomics can be beneficial. We automated the pipeline. Let's go back. Pipeline is automated. It creates huge data sets, and in our data we have observed almost a large part of the kinome. And how to make this actionable, boil it down into, let's say, a score that the oncologist can use. So we looked at all the tools out there. There's many tools, but none of them are single sample tools, or except maybe the PTM-SEA, but it's not so directly actionable on kinases. So we made a dedicated tool to score kinase activity. And we take the kinase observations and the substrate observations of the data that we combine into a single score that we can plot as a bar graph or as a kinase substrate network. We also make it available via web-based portal, and of course the formula or the tool can be also downloaded. And so for the substrate side, we make use of experimental data in Phosphocyte Plus, motive prediction from network-in, and then combine it with all the kinome from Manning. And with that, we only say a kinase is active if we have phosphopeptides from the kinase, and if we have phosphocyte information that can link to an upstream kinase. Only then, if we have it's a product of kinase observations and substrate observations or phospho data before we say a kinase is active. And this is to get you a flavor of how it works. This is an example of a cell line that has a BCR-ABL fusion. It leads to hyperactivity. This is known oncogene, single drug will work. This is the bar graph ranking within this sample. So here a single drug is enough to kill the cell. And this is an example of a lung cancer cell line with an ALK mutation. ALK is one of many hyperactive kinases, and you can understand that here a single ALK inhibitor will not work. And this cell line is very resistant to ALK inhibition, so you would need more drugs to kill this cell line. And this is kind of what we've now validated in multiple studies. We can rediscover known oncogenic drivers arising from fusions or amplifications or other chromosomal events, mutations in various different studies. And with INCA, single sample INCA, it always ranks in the top 3. But the more challenge is, can we predict when genomics cannot find a driver? And for this, we teamed up with Livio Trusolino in Italy, and we profiled a subset of his patient-derived xeno cohort that was extensively analyzed by genomic and RNA-seq analysis, and many resistant mechanisms for EGFR targeting therapy are known. So we profiled 30 PDX tumors, many with known aberrations, and then there were a few without aberrations. We did our single sample INCA approach to explain the resistance. We also did some group-based analysis where you can see that resistant tumors and sensitive tumors, they can be separated on all information layer, the tyrosine phosphoproteome, the global phosphoproteome, and the proteome. And it's easy to, let's say, to rediscover genomic drivers from amplifications or from mutations, and that co-targeting these with EGFR targeted therapy is beneficial. Same here, but here there was genomics didn't pinpoint any kinase activity or driver, and we find high insulin receptor activity and MET activity. So these are the PDX tumors, single sample Inca ranking organoids. We selected the drugs in red targeting kinase activities with the BMS drug, insulin receptor, the other one afatinib, EGFR. And we see that based on profiles selected, insulin receptor targeting therapy helps, and then the combination treatment is even better, and targeting a low-ranking kinase activity is not so helpful. And the same here for another model, not genomically explained. There was a lot of SRC and EphRIN activity, and then using dasatinib to target this is beneficial. And in high dose, when dasatinib also targets EGFR, let's say only dasatinib is enough. And this is now expanded in an ongoing consortium study that also includes clinical trial material. Same kind of team in T-ALL, where we had the genomic drivers can be easily picked up with single sample phospho plus Inca analysis. And then we also had genomically unexplained, where we also see high SRC family activity, but targeting only SRC family activity works in the genomically driven cancers, but the other ones need more than one drug. And what stood out was insulin receptor activity in a lot of the non-genomically driven cell lines, and then co-targeting insulin with SRC family activity is effective synergistic combination. And we also found this activity in PDX samples of T-ALL. And when there is, importantly, no target, no benefit of the combination, and when the target is there, it's either synergistic or additive. Same true for other PDX models, where we find YAC as a co-target in some of them, and combination therapy helps. Can this be done in clinical samples collected, for example, in a clinical trial? Yes, we downscaled the protocol. It can be done. Because sometimes we're fighting for needle biopsies, the genomics, proteomics. So we also have an all-in-one sample prep protocol where we know that proteome or phosphoproteomics can be done on the DNA RNA extraction leftover fraction. So don't throw away your leftover from DNA and RNA analysis. Here are some numbers that, so we are profiling now in several clinical trials, that most of the biopsies we could do, all of them we can do proteomics, most of them we can do global proteomics, and about 65% we can also do the tyrosine phosphoproteomics workflow. I'm going to skip this in view of time, but this is pre and on treatment and at progression samples, where, again, these Inca profiles, we see explanation for progression and differential activities in short progressors and people who live long on combination treatment. So to summarize this, single sample kinase activity ranking identifies known oncogenic drivers. It can help prioritize activities, and also in the absence of genomic drivers, it can find kinase activities for targeting. And the way forward we see is in the Netherlands we need a large-scale effort. So in the Netherlands we have the national basket study, the DRUB trial, and there's genome sequencing used to select patients for treatment baskets. We want to add the proteome and the phosphoproteome, but we still need to find the funding. This is just to highlight, in Switzerland and Germany, there are multi-omics precision oncology programs at a large-scale level. This picture comes from the Swiss commentary or white paper, and they plan to do or are doing many omics, and they have a 4-week turnaround time, because the question is, can this be done in clinical real time? I think so many omics is not practically, it would be very expensive, and 4 weeks is a long time. a better approach is the one Kuster is pioneering the way. He does, they add the proteome and the phosphoproteome to DNA and RNA, and they show it can be done. It can be done. They have now experience from the past few years where they profiled now almost 2,000 tumors. They have 2-week turnaround time. They have been doing this every 2-week cycles. Over 800 phosphoproteomics were discussed in the molecular tumor board, and it's sometimes in 30% of the cases it changed the treatment recommendation. So these are very impressive results. I think Bernard is leading the way to show that this can be done in clinical real time. And with this I will stop with the take home for more precision in precision oncology. We need multi-omics that includes the phosphoproteome. I would like to thank you for your attention and my team, collaborators, patients, and the funding. Thank you.
Connie, thank you very much. An impressive overview as a keynote should, but I really, really appreciate the messages. Are there questions from the room here? We have mics going around. Let me Peter, please.
Thank you for this. Does it work? Yeah. Thank you for this very impressive overview. So my question is actually, so how far are we, for example, when we look at phosphoproteomics and proteomics from prospective clinical trials where we really evaluate drug decision decisions in a real world setting?
In the Netherlands, not so far yet. It's all retrospective. And we did a lot of PDX in vitro, in vivo validating what we observe retrospectively. So I think that's where our strength is. In Germany, Bernard Kuster has examples where in the prospective setting, for example, it's anecdotal, because a lot of these patients never get the treatment recommendation because they cannot be financed or the patient is too weak or whatever, many issues or many challenges. But he has some examples where, for example, there is an oncogenic mutation in EGFR, and then there is a mutation of insignificant thing in PDGFR. Then with the phospho, they see actually EGFR is not active, but PDGFR is very active. So that changed the recommendation to the PDGFR targeting drug, imatinib, if I'm correct. And the patient was stable for a pretty long time on this drug, and then when the patient progressed, they put the patient on the other actionable mutation, and that didn't work, as would have been expected based on the phosphoproteome data, because it was not active. And there are, from this experience of these almost 2,000 profiles, of which half of them were discussed, and a handful of patients had actually the treatment. There's very nice anecdotal evidence, and this will be hopefully come out in Nature soon. It's under review. So we need, and they advocate also to do this in a trial setting, a basket trial, where, so now in the Netherlands we have DRUP that is based on genome profiles, and I think we need the DRUP Plus that also includes baskets with proteome and phosphoproteome profiles. We are ready for it. The field is ready for it. So now we have to find the funding. But the genomics colleagues, I mean, last week or 2 weeks ago, there was the clinical cancer genomics meeting in the Tropenmuseum in Amsterdam. There was the 10-year party of Hartwig. I went there, I was there, to celebrate. I mean, the genomics field is still kind of struggling to get the funding, and they are not ready. They don't want, actually. Or I mean, it's also a competition for money. But I mean, this is what needs to happen next. I think it's very impressive what in the Netherlands what Hartwig has achieved. That's fantastic. And also some of the genomics is now reimbursed by the verzekering, the insurance companies, especially for the CUPS, the cancer unidentified primary. and some other indications. And I think it will be, well, hopefully in the coming few years, that we can open some baskets within DRUP, for example, that are based on phosphoproteome profiles. And for colorectal cancer, I hope this can be a first basket, because we are very far in validation of a low-dose triple drug combo in colorectal cancer. So hopefully for that we can have a basket at some point.
Thanks, an extensive answer, but you made several points which probably had several questions addressed already. Are there any more questions from the audience? Let me ask your opinion on something you mentioned a few times. Technology is ready, now we should move to clinical application. You gave kind of a view on that, on the basket trials, but how can we accelerate that? Because I do agree. How can we accelerate that, all these technologies, maybe not the high end, but the lower end, still have clinical impact and are applied, if you like?
I think it's a matter of large-scale funding, because what Bernard did took several large funds. Like there was, Germany had a large consortium funding that was specifically to drive mass spec into the clinic. So mass spectrometry for precision medicine program that was 15 million euros per consortium. And there was money of his ERC advanced grant going in there. So I think he has 2 bioinformatics people, 1 person in the lab, 2 postdocs preparing these molecular tumor boards. So it's like a 6 FTE effort that he had on these molecular tumor boards thing. So it's, and you need, I mean, it went over a few years, so it's pretty significant funding what is needed. And I guess possibly, yeah, teaming up in the Netherlands, collaborating with different groups that can bring different aspects to the table could be beneficial too.
Cool, cool.
Consortia like X-omics.
Very good. I needed that last one. Very good. So let me thank you again and give her a big hand, please. So.